Okay, so here we are. We are doing lab 3, which is all about neurophysiology. Okay, so the nervous system. Basically, overall, our nervous system is in charge of communication. So the nervous system utilizes cells called neurons, and these neurons generate and send action potentials. So we'll talk about exactly how an action potential is generated and how it is conducted. But overall, we could say that these action potentials are they're electrochemical impulses. And when they are going through the body, they are sending signals to help us coordinate whatever needs to be done. So, um, we are not going to go into depth into like all the different types of neurons and then like neurons versus neuroglia, which are the support cells in this class. We are just going to simply go over the structure of a basic neuron since it is just one unit. So, what is a neuron? Well, here is just a basic, like cartoon version of a neuron. So we can just name the general parts and get an idea of what each part does. So, again, the role of this cell or this neuron is to basically generate, propagate, send these action potentials to other cells or other tissues using neurotransmitters, which are essentially chemicals, okay, that are going to go bind to different things. Okay, so let's see what we have here. So, starting up at the top, you can always notice that there is a nucleus, right? So, whenever we see the nucleus, we can generally kind of go around here and we could say, well, this is the cell body. Okay. Cell body obviously contains the nucleus. And also, what's more important is that surrounding the nucleus, we have these branch like projections that are called dendrites. So, dendrites are important because they are receiving signals. Okay. So, right here, all of these little branches receive signals from other neurons. So impulses are always going to enter this way and then be propagated this way. We never get impulses going this way. Okay, cool. So we got our dendrites. They are surrounding our cell body, which we were able to locate because we found the nucleus. Then, if we go down, we are approaching a very, very important area right here where I have this arrow. And this is blank for you to write in what this structure is. So here, right where the cell body ends and this tail called the axon begins, this is called the axon hillock. Okay, the axon hillock. And the significance of this area is that essentially we kind of gather all of the signals up here at the cell body and the actual action potential, if it's going to occur, will be generated at this area. Okay, so for now, just note that this is the axon hillock. Okay, so moving down the rest of the neuron, this one large projection, this is called the axon. So axons conduct action potentials or these electrical impulses away from a neuron. Okay, so it's like leaving this way. All right, so here's the axon. Now you'll notice that covering the axon, there are these little yellow pieces. These represent myelin, okay, or the myelin sheath. The function of myelin is to essentially insulate the axon. This insulation protects it and also helps to speed up the transmission of these electrical impulses. And we'll talk about this in a moment in depth. But for now, like I said, we're just naming the parts. Here's the axon. These little yellow pieces represent the myelin sheath. And at the very end of the axon, right here, these little end points are called axon terminals or axon end terminals. And here is where we're going to find neurotransmitters. So our little neurotransmitters or these chemical signals are going to be waiting here at the end of these axon end terminals, and they will be released if there is an action potential. All right, so those are the parts of a neuron. If we look over here, we can see two neurons synapsing together. Okay. Hopefully at this point, you know a synapse is a space, okay, which means that neurons do not physically touch. They get really close to each other, but they do not physically touch. So here's neuron 1, a presynaptic neuron because it is in front of the neuron. And then here's neuron 2, which is a postsynaptic neuron since it is after the synapse. And we can see that the neurotransmitters were released from the presynaptic neuron and they're able to float on over to the postsynaptic neuron. So this is how a signal can be sent from a neuron to another neuron. All right, so now before we jump into all the nitty gritty, let's first talk about the nervous system overall. So here you can see this essentially oversimplified version of the nervous system, but here we're just trying to see the difference between the central nervous system and our peripheral nervous system. So those are the two big parts. Let's first start out with the central nervous system or the CNS. This one is simple. It's just two things. The central nervous system is literally just your brain and your spinal cord, which we see here in purple. Okay. So it's in the center, right? So it's our central nervous system. And generally, what we say is the central nervous system is in charge of integrating and interpreting all of this sensory information that the body has to process every second of every day, right? So our body is consistently receiving input and the body needs to kind of make sense of that. So that would occur either in the brain or in the spinal cord. Now, everything else you see in this picture, everything in yellow represents the peripheral nervous system or the PNS. Peripheral meaning on the outsides, right? So it is on the outside of the CNS. So the peripheral nervous system, we could have a whole lecture on it, but we're just going to keep it brief since we are, again, one unit. What I'd like you guys to know about the PNS is that it contains our sensory neurons and our motor neurons. Okay. So sensory neurons, like the name infers or implies, sensory neurons bring info in, like your senses, like think your special senses. Okay. What you touch, taste, smell, hear, feel. Okay. Those are sensory neurons. They are bringing info in to your body, right, from the outside world for the most part. Bottom line being, they're bringing info from the PNS towards the CNS, which is the brain and spinal cord. Now, the opposite of that would be a motor neuron. Motor neurons, just think motor movement. Motor neurons take information away from your CNS or your brain and spinal cord. So motor neurons are going to basically travel outwards, and usually our motor neurons are going to be innervating a muscle. And that should kind of make sense, right? Your brain or spinal cord could be sending a signal to a muscle in your arm telling it to contract. Okay, so that is info leaving the CNS to make something occur, some action. All right, so CNS versus PNS. All right, so now let's look at the three different types of synapses, which again, they're just gaps, they're spaces, three different synapses that we can have. First off, we've kind of already mentioned this one, but neurons can talk to other neurons, okay? And we can see here they're not physically touching, okay? They're just right next to each other, but essentially this yellow neuron would be communicating with this purple neuron. So that's a neuron to neuron synapse. The next one we could have is a neuron that is innervating or trying to talk to a muscle. So this, we'll talk about muscle in another lecture, but this represents skeletal muscle. Okay. So here is skeletal muscle, and we have a neuron that is reaching out and doesn't look like it, right? But they're hovering over certain parts of the skeletal muscle. Okay. So basically, probably telling it to contract. Lastly, we can have a neuron communicating to a gland, right? So here, it's a little bit hard to see, but these are neurons in yellow, and they are sending a signal down to a gland right here, which is called our posterior pituitary. This is just an example. But yeah, so a neuron can send a signal to a gland and tell that gland to secrete a hormone or whatever it may be or another neurotransmitter, but basically it can give it a signal. So bottom line here is that there are three different types of synapses. We are going to focus on this one today, and then this one later in another lecture. This one we'll briefly talk about, but these are the two big ones we'll go over in this class. Key being that we need these neurons to essentially release their neurotransmitters, okay, or their chemical signals, so that those neurotransmitters can travel and bind to whatever tissue or cell that they're trying to talk to, and then get it to do what it's trying to have it be done. I don't know. Basically, it's just communicating something to that tissue, okay, whether it's like contract or release this neurotransmitter, whatever it may be, it's a signal being sent. All right, so now we are going to talk a little bit about gradients. And we talked about this a lot last week when we talked about the cell membrane and how it behaves and how we can have concentration gradients based on different types of solutes and how they behave. So for this lecture, we're mainly talking about ions, movement of ions across the plasma membrane. So we'll just, again, remember, we know what a neuron actually looks like, right? Like this. But we're just going to pretend like down here, we'll say, is the neuron. Out here is outside the neuron. And then this would be the plasma membrane of that neuron or the cell membrane. So um, there's two parts we need to kind of take into consideration here. We have an electrical gradient that is made essentially because we have a stark difference in positive and negative charge across the membrane. Okay, so usually one side is more positive, the other side is more negative. Okay, and there's a reason for this, which we'll talk about. So we do want to have some sort of electrical gradient here, which is essentially a difference in charge. How that gradient is actually formed kind of has to do with the concentration. Here, in this specific lecture, we're talking mainly about sodium and potassium, and these are ions, right? So these are also solutes. So sodium and potassium, they are going to be moving in and out of the cell and kind of creating a concentration gradient and an electrical gradient, sorry, an electric gradient. So this kind of imbalance of these particles is what is going to create either a resting membrane potential or an action potential. And we'll talk about what that means in a moment. But for here, just want to start with, let's see here. So if we combine these two together, electrical and concentration, that gives us something called an electrochemical gradient, right? Which means we are considering both the concentration of the solutes and also the charge that those solutes are imposing on that membrane. So for example, let's say over here, um, the green balls represent potassium. And hopefully you're thinking a potassium ion is charged, right? As a positive charge. And hopefully you can recall that things that are charged, like ions, cannot freely cross our membrane. Okay, they cannot freely cross. They require a channel, right? So here we have channels, these green channels, and right now they are shut. So at this point, all of the potassium is stuck inside this cell and it cannot leave. Okay. So basically it's creating a positive, right? That means that over here, we have all these positive ions and none out here. And we have all this potassium out here and none out here. So it's creating a chemical and an electrical gradient across this membrane. Now, what we could do though is let's say we opened the channels. Okay. Let's say there is something that triggered these channels to open, and now there is a way for potassium to leave. And hopefully you can deduce that right now, potassium is super crowded inside the cell. It does not like to be crowded. It would prefer to spread out. So if these floodgates opened, it would simply diffuse out into the extracellular environment. And that's literally what would happen, right? So basically, this is just kind of setting us up for what we're going to see in terms of the movement of ions in the neuron. Okay, so enough of this like general talk. Now let's just talk straight about what happens at the neuron. So here is, again, we know neurons don't look like this, but this represents the neuron's plasma membrane. So inside the neuron, outside in the extracellular environment. So we see here that this I guess situation is representing an environment that is negative 70 millivolts, meaning if you put a voltmeter inside of this plasma membrane, the voltage would read negative 70 millivolts. Okay, so negative voltage. So let's see what makes this negative 70. Well, there are two colors here. So the pink over here and the green over here. So the green represents potassium and the pink represents sodium. So right now, we can see that there is more potassium inside the cell as opposed to outside. But there is more sodium outside the cell than inside the cell. Okay? So that's a situation chemically. And when that occurs, okay, when there's more sodium out and more potassium in, like I said, if you measured it, it would read negative 70 millivolts. That is the difference. So when we're talking about neurons, it's really important to remember how they exist in their resting state. Like, what is their preferred state? So to remember this, you always want to remember that this neuron, okay, you could think of neurons like islands. Okay, so inside or on the island, for example, here you can see we have trees, and usually islands have banana trees. Okay, this crazy analogy, go with it. It'll help you remember. So hopefully you know bananas are made of potassium. So that means there's more potassium inside or on the island. Now, sodium, salt, right? Islands are usually surrounded by salt water, so surrounding the cell, that means that it's going to be higher in sodium, like the salt water. Okay? So remember that. That is the situation we have when we have a negative 70 millivolt reading on our voltmeter. Okay, so how do we maintain this though? Okay, so this is how cells are normally. Okay. But there's a couple of things we need to consider here. So here's the same situation. Okay. We have our cell, more potassium in, more sodium out. So we have already talked about the concentration gradients. That check, we got that. But we have not talked about the permeability of these solutes, essentially, right? Or these ions. So it's important to remember that, for example, here, this green, this is a potassium channel and this is sodium channel, meaning only sodium can go through here and only potassium can go through here. These guys, the potassium channels, are very, very leaky and they are more permeable than the sodium channels. If something is more permeable, that means that those ions are going to be able to flow through more easily and thus kind of faster, if you will. So just keep that in mind. At rest, more potassium is going to be able to leak out than sodium is able to enter. Okay, so just remember, leaky potassium channels. Okay, so if that occurs, right, if potassium is just actively leaving the cell just because it can, right, it would like to go down its concentration gradient. If there's more inside, less outside, and the door is open, it's going to go ahead and flow on out, right? Think of it like a cracked door, okay? Things can get out, but slowly. So what happens over time is if enough potassium leaks out, then we're losing a ton of positive inside that cell. So it's going to mess with the concentration gradient. It's going to make us lose that negative 70 millivolts, which we really, really want to keep. Okay, I'll tell you why in a moment, but we really want to keep the voltage at negative 70 or resting membrane. So what we have to do in order to maintain that is we need help from the sodium potassium ATPase pump. Okay, sodium potassium ATPase pump is obviously critical and it uses energy or ATP. So what this guy does is kind of help us to combat this loss of positive. So you can see again, it's color coded. This pump takes 3 sodium and pushes it out of the cell and takes 2 potassium and puts it into the cell. And by doing that, or in order to do that, it requires ATP. So taking 3 sodium out and 2 potassium in, that requires an ATP. So all the time, we are using ATP just to maintain this gradient. Why are we needing energy for this, right? Like, where's the active part of this process? Well, if you think back, okay, over here, if you remember the island analogy, we have potassium. It is already high inside the cell in concentration. So if we're trying to push more potassium in, that's against the concentration gradient. And then if you remember salt water on the outside, sodium is higher. We're pushing 3 sodium out. Sodium doesn't want to go out. The concentration's already high out here. So we're pushing these ions actively against their gradients, and thus that's what requires ATP. Okay, so here it is in this like basic schematic, and then also over here, same thing, just over here is the inside of the cell, outside of the cell, and just explaining how we maintain that gradient. And of course, Kendrick doing, you know, doing his thing over here. Okay, so now let's talk about how do we actually generate these action potentials, right? We've kind of talked about a little bit about them. Why are they important? Well, they're going to help us with signaling. So first thing I want to mention about an action potential, or how to generate one, is that an action potential can only be generated if the voltage of that neuron hits threshold, which is negative 55 millivolts. And right now, we've just been focused on maintaining a voltage of negative 70. Okay, why? Well, in order for us to actually even think about hitting an action potential, we need the membrane to be at its resting baseline, which happens to be at negative 70 millivolts. Okay, so at all times, while neurons are waiting for a signal, this is where you want them to be. All right, so let's talk about it. Here, let me explain this. So on this graph, we have time, okay, and then we're measuring the membrane potential or like the voltage, right, of the neuron, and of course, millivolts. And this is color coded according to the like little spikes on the graph. So what we have here are graded potentials, EPSPs or IPSPs. An EPSP, or the green ones, these are excitatory postsynaptic potentials, meaning they send an excitatory signal to the neuron, okay? Like they're telling it go. They're encouraging the neuron to go towards threshold. An inhibitory postsynaptic potential, like it kind of sounds, right, inhibits the neuron from getting towards threshold. Okay, so it sends like a no-go signal. Okay, and that's represented by this like pinkish red color. Okay, so let's kind of go through how this works. Right here, say we're measuring right here at this axon hillock, right? Remember, that's important because this is where the action potential is going to be generated if we hit threshold, which is negative 55. So right now at 0 seconds, nothing has happened. It's at negative 70, that's rest. But then we get an EPSP or an excitatory signal. So we'll say it was this neuron over here, okay? This down here, hopefully you're able to visualize that this is an axon N terminal of a presynaptic neuron, meaning you can't see it here, but there's another neuron that is sending a signal to this neuron. Okay. And it's sending a signal at this dendrite. And that signal is excitatory. Okay. So it's saying like a go, which means it's going towards threshold, which is here. Okay. But we did not reach threshold, so we do not get an action potential. But then a second neuron comes over here and it sends its own excitatory postsynaptic potential. So that's great. We got another one. So we're getting closer to threshold, right? But we still haven't made it. So now time is passing and we get a signal over here from this neuron. This one is red. This is an IPSP or an inhibitory postsynaptic potential. So you can literally see now the voltage is dropping because it is pushing it away from threshold. Okay. Then some time passes and a third excitatory signal from another neuron gets sent at this dendrite, and we're getting again trying to get closer to threshold, but still not quite. Then over here we get another signal, say from this one. It is inhibitory, so it brings us again further from threshold, but eventually, I think it was this one. I can't remember which one, but anyway, a different neuron fired an EPSP, an excitatory potential, and that one finally did the job, right? Because this one finally hit -55, which was threshold. And you can see we get this huge response, like this increase, massive decrease, and then back to rest. What we just did right here at B, that's the action potential. Over here at A, this whole like go, no go, go, no go, that whole like kind of fight situation, that is summation. Okay, we are summating all of these different signals and trying to get an action potential essentially. Okay, and like you can see, it did not occur until this point in time because that is when the actual voltage hit. Okay, so basically you just have to recognize here axon hillock, that's where they're generated, but they are only generated, they being an action potential, they are only generated when we hit -55 millivolts or threshold. Okay, all right, so at this point we have created an action potential. So let's stop and do a quick question here. If we have a signal and it reaches -60 millivolts at the axon hillock, what is the next step? Like what would happen? So you can pause, take a moment to answer, use this graph if that helps you. Okay, so you can see here that we are starting at rest. And then it says the signal is going to reach -60, which would be right here, right in between -70 and -55. And hopefully you realize that -55, right, threshold, it didn't make it, right? -60 is less than -55. So there was not enough voltage to get an action potential. Okay. So even if you didn't know what these meant, okay, you will in a couple of slides, you should say no action potential occurs. Okay, if you don't hit threshold, nothing occurs. That neuron is just remaining at rest. All right, so here it is, the action potential. Um, Again, what is it? It's really just a really rapid, brief, all or none response, okay, that's going to eventually get propagated or sent down a cell membrane of some type of cell or tissue that can be excitable, okay? Like a muscle, okay? It could be, like we said, it could be another neuron, could be a muscle, could be a gland. So, what, um, how do I want to say? Okay, so the thing in red right here, this is the actual action potential, okay? And it always will be, like we said, it has to always be, sorry, it only occurs when we hit threshold, which is -55, always, okay, millivolts, all or none just means that either it hits it or it does not. That means there is an action potential or none. There's not going to be like a mini action potential, right? Um, we're going to use these voltage gated ion channels to essentially create this action potential, which should make sense, right? We are talking about charged or, yeah, essentially charged ions, right, moving across the membrane. So we are talking about a channel that is triggered by voltage, hence a voltage gated ion channel. Okay, that means that channel opens at a certain voltage. And then um, also, these action potentials, they do not die out, right? Like it's going to travel all the way down an axon and maintain its height, right? Or maintain its voltage, because that would be the height. And we'll talk about what that looks like in just a sec. But for now, okay, there was some triggering event that triggered an action potential. And then after we get this like kind of refractory period, and we eventually get back to resting to get another action potential. So an example or like the best way I can kind of help you visualize this is if you've ever been to like a baseball game or some sporting event and they're doing the wait, right? So somebody starts it, okay? Everyone's at rest, but then somebody decides, okay, we're going to start this, and they make everyone in their row stand up, okay? So that, when everyone stands up, that chunk of that section, that's called depolarization, okay? And then what happens, right? You sit back down, okay? When you sit back down, we're going to call that repolarization. And it's going to keep going forward, right? We don't go backwards, okay? The wave goes around the stadium, at least that's the goal, where everyone is standing and sitting, okay? Okay. Okay. So here are the parts of an action potential. It's kind of a lot, like it looks a little scary. There's a lot of stuff on the slide, but I find that it makes a little more sense when we see it as a big picture. So kind of think of it like a little story here. So again, we're measuring voltage, right? We're trying to measure voltage in the cell membrane over time. And action potentials are quick, very, very quick. So that's why we have time in milliseconds. Okay, so let's just start out with blue. Here we are at -70 millivolts. Okay, our neuron is just sitting there, resting, waiting to be stimulated. Okay, so it's resting at -70, just minding its own business. And remember, the reason that it's at -70 is because there's more potassium inside and more sodium outside. Okay, so don't forget this. Then, at about 1 second, we get one signal. Okay, this signal, hopefully you're thinking, was not enough, right? It did not hit threshold. Then, okay, so nothing happened, right? We got another one. Still did not hit threshold, so nothing happens. Third signal comes in. This one, this green, it does hit threshold, right? Its amplitude was high enough to hit -55, and this is kind of like where we get started. So hitting threshold triggers something called depolarization. Okay, we're depolarizing the membrane. What does that mean? This means that all of the sodium channels on the cell's membrane are going to open. Okay, so remember these voltage gated sodium channels? When we hit threshold, those channels open up and sodium is going to rush inside of that neuron. Okay, so that's what this represents here, this whole green, like increased part. Okay, so here we have sodium rushing into the cell. And hopefully you can picture that. Well, the cell at rest had a bunch of sodium outside. And then we opened a gate that was only for sodium. So sodium was like, okay, it's really crowded out here. I'm just going to take this opportunity to leave where there is less of me. Okay, get away from the rest of the sodiums. So it does. So that is why it rushes into the cell. Okay, down its concentration gradient, no energy. Okay, so that's going to keep happening, keep happening, keep happening until we reach about a positive 30 voltage. At that point, those sodium channels are going to snap shut. Okay, so no more sodium is moving. That's the peak of our action potential. Then we are going to talk about our potassium channels. So at this point at the peak, potassium channels open. Okay, so there's a bunch of potassium channels on the membrane. Now it's their time to shine. When they open, you have to think about, well, they're only for potassium. Where is potassium going to want to go? If you go back to where it was at rest, potassium was super high inside the cell. And it doesn't like that, right? It's crowded. So if it has an open door, potassium is going to want to leave. So that's what it does. When potassium channels open, potassium moves out of the cell. Okay. So here, this yellow piece right here, potassium is leaving. Okay. The thing is, these potassium channels, like we said earlier, they're like leaky, they suck, they're slow. Okay. Because they are slow, we have kind of a little overshoot here, right? So let's talk about this here. Potassium's leaving, 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 leaving. That's great. The action potential is complete. But we have now surpassed resting membrane potential, right? Remember, we just want to get an action potential and go back to rest, right? Stand up, sit down, like in the wave example. Because if we surpass negative 70, we can no longer get another action potential at that neuron. And that's a problem. Okay, we can't just get one signal and we're done for the day, right? We need multiple signals multiple times a day, okay, like infinite. So we've got to fix this. So this down here, when we're in this period where we're beyond resting, this is called hyperpolarization, right? We're beyond polarized. And this is again because potassium is still leaking out of the cell, okay, causing this excess, causing this refractory period. And that means no action potential can occur, okay, until here. So what happened? How did we get from down here back to rest? That's our sodium potassium ATPase pump. Okay, remember that pump was responsible for maintaining our gradient. Okay, what does it do? It throws 3 sodium out and 2 potassium in, and that uses ATP over and over and over and over until we have officially reached negative 70. And now that neuron is happy and ready to get a new signal. Okay, so that is essentially the action potential. Okay, I love this chart. I love that it's all like complete on one little concise area. Okay. So things I could ask you are like, obviously not have the words here, but say, what channel is opening here? Or over here, I could say, the ion potassium is moving in or out of the cell. Or who's in charge of reestablishing the gradient here? Okay. Things like that we could ask you. So you got to know at what part of the graph are these things happening, right? Okay, so this is all the stages of an action potential. Especially in your homework, make sure you are understanding what's happening on depolarization, repolarization, hyperpolarization, back to rest. All right, let's try another question. So, what type of membrane transport is at play when sodium rushes into a neuron? And here we're saying sodium is these purple balls. So you can pause it, give yourself a minute. Okay, so if we look at this photo, inside, we can just say whatever cell this is, let's say it's a neuron. Uh, how much sodium is in the cell versus outside? Well, you can visually see that there's more sodium outside than inside. So if sodium is rushing into this cell, that means that it's going down its concentration gradient. It's going from high to low. And we see that it's using a channel. And even if you didn't see the channel right, it says sodium. Well, I guess, yeah, actually does. It says sodium ion here. Ions cannot cross the membrane like this, right? They cannot. They need help. So that tells us that we are looking at number 3, facilitated diffusion. Okay. No ATP, so can't be active transport. Simple diffusion, that would be moving straight through the membrane. Can't do that. These are ions. Osmosis, that's the movement of water. So 3. All right, so let's get back to it. So we have now talked about resting membrane potential, summating, generating an action potential. Now we need to talk about moving that action potential or propagating it down a neuron. Okay, because just making an action potential and having it stay stagnant doesn't mean anything, right? The whole point is for it to leave and go tell something what to do. Okay, so let's talk about propagation. So propagation just means moving forward, right? And in this case, it's in one direction. Okay, action potentials do not go backwards. Okay, hopefully, right? If they do, that's a problem. So we're going to talk about how we maintain that unidirectional movement. So one thing that we have in place is something called the myelin sheaths, right? We talked about that earlier. Right here, this is like, so if you look in this box, we are looking at a zoomed in portion here. So the blue represents the axon, and then the yellow that's surrounding it, that's the myelin. So what is myelin? So myelin is like a fatty insulator, okay? And it coats the axon, okay, not the dendrites, right? We're talking about this tail-like structure, the axon. It coats parts of it, okay? Not all of it, okay? Parts of it. And this is to help, like I said, insulate the signal, help us to get that action potential down without losing energy. So how does that happen? Well, if you look in between two chunks of myelin, there is a piece of axon that does not have myelin. Okay, I like to say it's like a naked axon, right? It has a jacket here, nothing here. So this is called a node of Ranvier. So the nodes of Ranvier are the unmyelinated portions of the axon. And this essentially, like what makes it a node of Ranvier? Well, it has a very high concentration of sodium ion channels. So hopefully you're thinking, okay, why sodium? Well, it has a high concentration of these specific channels because those are the channels that we use when we're doing depolarization, right? Which is sodium channels open, sodium rushes into the cell. So sodium's rushing into the cell, hence you could see it's becoming more positive. Okay. So action potentials, once they're generated, and they're trying to, right, they're generated here, axon hillock, and they want to move down the axon to go innervate another neuron in this case, they will do this. Okay, the action potential will jump. So it will be, you can see here, depolarization, repolarization, jump. Okay, we do not get action potentials on the areas where there's myelin sheaths. Okay, and that's kind of nice. Okay, because if you look here, this one has no myelin. The signal's very slow and dies out. This one has myelin and it's very fast and efficient. That's because we get to skip all of this surface area, jump and skip. Okay, this like apparent jump of the action potential, it's called saltatory conduction. Okay. So, saltatory conduction means that action potentials jump from node to node to node as they are propagated down an axon. All right, so I do want to mention one quick clinical fact here, and that is I would like you to take a moment to think about a specific condition, okay, where somebody's body decides that it's going to attack its own myelin. Okay. It's going to just eat away at this fatty covering that is helping us to insulate our neurons. You can pause it if you want to think longer. But the condition that I'm talking about is called multiple sclerosis or MS for short. So MS is an autoimmune condition where, again, we don't know the reason why. Okay. Autoimmune conditions have different triggers, and sometimes we don't know what causes them, usually genetics, right? Something passed down, sometimes just bad luck. But essentially, and for some reason, this person's body decided that it does not like myelin and it's going to destroy it, even though it's very helpful for us. Okay. So, I don't even know how long ago, but back in the day, I'll just say MS was kind of like this is kind of like not a death sentence, but there's like nothing you can do. Like they were just like, that's it. Like you're just going to deteriorate. Good luck. Now, as science is advancing, right, we're getting treatments where we can actually I don't want to say stop yet, but slow or halt the progression of this degradation of the myelin. So we're on a good track, right? We're hopefully getting somewhere. When I say we, I mean the researchers, not me. But hopefully we'll be able to stop this. Okay. So I would like you to know MS, multiple sclerosis, is an autoimmune condition where somebody's body, for some reason, attacks and eats away at the myelin on these neurons. And it's the myelin specifically in your CNS or central nervous system. So think about what kind of symptoms they might have. Okay. It's very broad. Okay. And it varies from patient to patient because it just depends on which area is being affected, right? Let's say um, it is a neuron that has to do with your eye, okay, called your optic nerve. Well, if that neuron is having issues, then you will get something called optic neuritis, okay? So if it starts getting attacked, I don't want to say issues, if it gets attacked. So that's one signal. If somebody suddenly gets optic neuritis, they would look to test them for MS, okay? Poor coordination, numbness, tingling, like brain fog, fatigue. And this is not just like one day, right? These are persistent for like days, okay? Then they would definitely want to test you for MS. How do they do that? Spinal tap, okay, is how they actually definitively diagnose, but they're also going to do obviously like an MRI as well. Okay, so just a little fun fact MS. All right, so let's finish up our action potential. So once that action potential occurs, okay, there is a period of time where we literally cannot get another action potential. Okay, and that's called an absolute refractory period. Okay, so if you look here, we have resting, some event, depolarization, repolarization, surpassing, right? We are surpassing rest. So if we are not at, sorry, over here, if we are surpassing, okay, we shouldn't be able to get another action potential, right? So we call that a refractory period. And there's two types. Absolute refractory period means it is impossible to get another refractory period because they are all or nothing. So while one is occurring, a second one cannot occur at that exact same time. Just cannot, right? They cannot take up the same point in time. But let's say, like over here, if we are just like a little bit below -70, and let's say we get a really big excitatory postsynaptic potential, right? A big EPSP, and it's able to push the voltage up through -55, okay, then we could get one, but it's hard. Okay. What that means is it is relative because it's not ideal like when you're at rest at 70. It's a little tougher because you have to overcome that extra um, I guess like that extra voltage. Okay, so very hard to do, but not impossible. All right. So why do we have this? Well, again, we want to make sure that the action potential is going forward in one direction. We don't want to like stop and move backwards. Okay, signals need to go forward. So that is essentially why we have this design. Okay. All right. So tying things together. So, kind of going back to the beginning when we talked about sensory neurons, motor neurons, CNS, PNS, here is the big picture. And this is going to be important for this lab. So, sensory input is from our sensory neurons, right? Like what we see, taste, touch, smell, etc. Okay. Everything from our environment, that is sensory input. All of the sensory input gets sent to our CNS, right? Our brain and spinal cord. So it can be integrated. Here's where your body kind of decides like what is a priority, which signals are most important, which can I like filter out, and what are we going to do next, right? That's what's happening in the CNS. Okay. PNS, okay, peripheral nervous system, is when, let's say, your body decides we're going to contract this muscle. Okay. That would be motor output when you actually contract and move that muscle because you're using a motor neuron. Okay. So PNS, remember, is going to be like sensory neurons going this way. Motor neurons going this way, meaning towards a muscle or an effector that's going to carry out some action. Okay? Sensory input, integration at the CNS, motor output, some effector. Okay, so basically, obviously you're not doing this at home, but you could if you had something similar to the little mallet, um, the reflexes. So we are going to test two reflexes, and I'll talk about those in a moment. But you probably already know what's going to happen. You've probably had them done before, so you can use your imagination. But essentially, we just want to touch on why do we have reflexes? Okay, so, or what are they, first of all? So, a reflex is an involuntary motor response, meaning it's a movement that you cannot control. It is out of your control. And that movement is triggered by some stimulus. And because it's out of your control, that means that the signal doesn't go to your brain. Otherwise, you would be able to stop it. So, where does the signal go? Well, it means that some sensory neuron, okay, like for example, this blue one, it's sensing something over here in the muscle, okay? We'll talk about what in a moment. And it senses danger at the end of the day. And it sends a signal to the spinal cord. This is like a little chunk of spinal cord. And that spinal cord, okay, obviously you don't know this, right? You cannot have awareness, okay, of what's going on in your spinal cord. But we get an automatic response leaving the spinal cord via, you can see in red, motor neurons that are going to tell some effectors to do something, which in this case is contract or relax to protect you. Okay, so they are protective responses that you cannot control. Okay, and obviously these are just important to protect your body, right? Like you, just like so you don't tear your Achilles or tear your ACL, like all of these very extreme situations, your body does its best to protect you from them. Again, does its best through reflexes. So those are reflexes we still have as adults, but there are others that you kind of lose, right? Like when you're a baby, you have more primal, like survival instinct type reflexes, like the suckling reflex, like latching on for milk, or if you've ever like put your hand near a baby's hand or just your finger, they'll automatically grasp. Okay, that's like a primitive form of protection, like holding on to whoever their caregiver is, um, learning how to crawl, etc. These are reflexes. They're not taught, or you can't teach a baby how to drink a bottle or how to breastfeed. So those are other examples of reflexes that you essentially lose over time. All right. So the two reflexes that we would have done in class are first off this guy. So this is the one you always, not always, but you've probably done at like your physicals or whatnot. And this is when you like dangle your legs off of the table and like relax, right? You really relax your legs. Then the doctor, whoever, comes up with a mallet and whacks you in the knee with it, okay? Right under your kneecap. So I don't know if you ever thought, like, well, why are they doing that? Okay. So hopefully you like kicked out, right? You kick your leg out. You cannot control that, right? You are just sitting there, they're just relaxing, and they hit you in a certain spot and your leg kicks out. Why? Well, essentially, what is happening is this mallet, if they hit the right spot, they are hitting your patellar ligament. Okay, so it's right here. This ligament holds your kneecap to your shin bone. But on top of your knee bone, or your patella, okay, it's your knee bone, you have muscle, okay, or your quads, okay, your quadricep muscles. So when you hit here, that force transfers all the way up to the muscles and you get a stretch. And that stretch, okay, if deemed too much, right, if they hit it hard enough, that stretch will be um, a signal, okay? So that stretch in the muscle will send a signal to a sensory neuron, right? Because it's a sensation. And that sensory neuron will go to, sorry, I can't really, here we go, the blue one, this one, to your spinal cord. Okay, not the brain, to your spinal cord. And then it will send two signals out. One motor neuron is going to go to the quadriceps and say contract. And the reason it's going to tell this muscle to contract is so that way it releases that stretch or that tension. So you don't accidentally snap this tendon, right? It's to protect you. That means there's too much tension, too much stretch. You got to change the position. At the same time, another neuron, this one, oh, it's this dotted one I see, this one, is telling your hamstrings, okay, on the other side, okay, like the back side of your thigh, to relax. So when the back side relaxes, the top side contracts. That makes your leg straighten out, and that's why you kick. Okay, so that's one protective response. Another one that we're going to try was the back of your ankle. If you just touch the back of it, okay, not your heel, like touch your heel bone and then go up. Okay, you'll feel this like bouncy, kind of straw like structure. Okay, that is your Achilles tendon. Okay, if you remember, I don't know, you guys are kind of young, but Kobe, okay, Kobe Bryant was on the Lakers, right? Superstar, amazing Mamba. He actually ruptured his Achilles on the field, on the court, and still shot free throws. Kind of crazy because, check this out. If you tear this tendon, you can no longer move your foot. Like it's like useless, okay? Because this is gone. It's like a levy or a lever type system, okay? So kind of wild. Okay, most people do not get up when this happens. But anyway, so what this right here, what we're going to do is take that mallet and like whack it. And what we would have seen is a quick little like kick, like if you're pushing down on a pedal, but it would be very, very brief. But essentially, what that does, again, is releases like that tension, okay? Prevents this tendon from snapping by moving the ankle kind of down so that way we don't get a tear. Okay, just another fun little reflex. Okay, what you guys need to know, okay, is this right here. So the knee one, where you are tapping, okay, it's right below the kneecap. You're specifically hitting the patellar ligament. Okay, it is a ligament because it is bone to bone, bone patella, bone tibia, and the muscle that contracts as a result are your quads. But there's actually four muscles in your quads, okay, like the front of your thighs. I would like you to know one of them, which is quadriceps femoris, okay? Then at the ankle, where did you tap? Okay, you should be tapping, like I said, the Achilles tendon. That's a true tendon. It is attaching bone to muscle, okay, muscled bone. The name of that muscle, there's two muscles, but we're just going to do the most superficial one, which is gastrocnemius, okay, or your calf. Okay, what you think of as your calf, that muscle, that's its name. All right. The other reflex we were going to do, which you can still do at home, you could just use like your phone flashlight or something, was the pupillary reflex, where I guess you could do this in a mirror or have someone else do it. You're going to shine a light in one eyeball very briefly, right? Very briefly, because it's going to really hurt you. and you are going to see what happens. Okay. And essentially, hopefully, if you're not like on any crazy drugs or I don't know, something, you should have this reflex. So when you're in like normal light, okay, like it's bright outside or you're in a lit room, your eye, okay, here's like the white of your eye, colored part, which is the iris, and then this black part, that's your pupil. Okay. The pupil is literally a hole. Okay. You cannot, like, it's not a thing. You cannot touch it, right? The muscle has a hole. It's a circular muscle, and that is the pupil. The pupil looks bigger or smaller when the muscle, the iris, or the colored part of your eye, contracts or relaxes. So when you shine a light, okay, so you start like this, and then you shine a light. Um, well, I guess it would be more like this. Yeah, you would start more like this, normal light, okay? Hopefully, if you put light in the eye, your pupil will get smaller because your iris will constrict or make that hole really, really, really small. And hopefully, you're thinking, well, it's got to be some type of protection, right? And yes, it is. It's because we do need light. We want to get light inside of our eye. That's how we see. But too much light damages the retina, damages our eyes, right? And like if you look at a light for too long, it like temporarily blinds you, right? So we don't want to have to think about this all the time. So our body does it for us as the pupillary reflex. Um, again, you can do both eyes, like you can follow the procedure in the manual and you can see a lot of things, right? You can see if um, they should both respond, right, at the same time and see if they're equal. Maybe one's bigger than the other, or maybe one responds, one doesn't. Um, you can also like look at the diameter and just, get some information about what's going on. But hopefully for all of you, you are healthy and well, and you just get this response, right? Where you get the iris contracting. All right. The last part of this lab was the 2-point discrimination test. So the point of this test was essentially you're going to use these like little calipers, okay, like this. These look really scary. But basically, it's just a little tool with 2 points. And this piece here, the black piece, you slide. And then there's a ruler. So you can measure distance. So you're measuring like distance here. And you want to measure the smallest distance between 2 points where you can accurately say, I felt 2 points. Like you felt the two little pinpricks. It's harder than it seems. And also, you obviously can't see while they're doing it. Okay. I think when we come to our next class, I'll um, either have you guys do it or I'll demo it so you can just see it before the exam. But basically, your partner, okay, would start like really big, okay, far away, and then gently touch your skin, either on the palm, back of hand, fingertips, neck, upper arm. And you would tell them one or two. Like I felt one poke or two pokes. And they would just keep getting smaller and smaller and smaller until you said you only felt one, but they actually touched you with two. Okay. So basically, we'll talk about like what the results mean. Um, this right here is just like skin. Okay, here's skin. Here, there's two areas. Okay, this is one neuron and another neuron. And each neuron is responsible for its own patch of skin. So if there's one point over here and one point over here, and you push down, you get a signal from this neuron and this neuron. And in your brain, you would say two. However, over here, if you get smaller, okay, and you actually get both points in that same diameter, where there's only one neuron, even though they're touching you at 2 points, because those 2 points are in the same area, your brain perceives one. Okay, so that would be your 2 point threshold. Wherever you say one, but they touched you with two. Okay. I know it's a little hard to visualize, so we'll go over it when I see you next. And maybe I can post a video of other classmates too. We'll see what I could do. Okay, so here is basically the physio behind it. Like this is what you would need for the exam. So what we're doing is testing your cutaneous receptors or receptors of your skin. Each neuron, each sensory neuron, like I said, has its own area of skin, and that's its receptive field. Okay. And depending on where you are in your body, the area of that receptor field can either be really small or really big. And that area is going to be inversely proportional to how many receptors are in that area. Okay, so it sounds like a lot of words, but this is what it means. So let's say you do the experiment and you get a really small 2 point threshold, like let's say 2 millimeters, okay, or 1 millimeter, you might even get. What does that mean? Well, that means that at 1 millimeter, you were still able to detect 2 points, meaning there was two neurons like that close together. So that means there's a high density of sensory neurons in that body part. For example, finger, right? Your fingers are very, very sensitive. You are able to detect things by using your touch, right? That's survival for humans, okay? So you need to have very sensitive fingertips and hands, and even the palms, right? To discern like what are you touching, what are you doing for survival, okay? So what does that mean? That means there's many receptors, but if there's a lot of receptors in that area, they're very tightly packed, and each receptor is only responsible for a tiny, tiny little area. That's what makes it so precise. And if that's the case, then that means your brain, okay, has a lot of sensory cortex associated to that body part. Meaning, right? So your brain has certain areas that are dedicated to each body part, okay, to perception of that body part. So basically, if you have a lot of cortex for that body part's very sensitive and you are very good at discerning small changes like hands, lips, tongue, toes, like very sensitive areas. Okay, now the other hand, if you got a really large 2 point threshold, okay, really big, that means that there's a low density of sensory neurons in that body part. Okay. So think of things the like your back, right? Back of your arm. Those aren't very sensitive areas, right? That's because there's not a lot of sensory neurons there. So if there's few receptors, then that means each receptor is in charge of a bigger area. And they're not going to get it right every single time, right? They're in charge of a huge area. They're going to be less accurate. With that, that means there's also a smaller portion of your sensory cortex or your brain associated with perceiving that body part. And that should make sense, right? It's not that important to consistently feel your back, right? Or the back of your arm. It's more important that you put your brain power towards feeling things with your hands, right? What you do every day, what you need to be precise. Oops, okay. So I want you to bring your attention to this beautiful man right here in the middle. Remember him, okay? His name is Homunculus. He is a sensory homunculus. Gorgeous, right? He represents the amount of sensory cortex devoted to each body part. So the bigger his body part, the more brain power, I guess you could say, you have towards that area. Okay. So, like, notice cheeks, lips, tongues, hands, feet. Those are humongous. So, they're very sensitive and they have a small 2 point threshold, right? Then the rest of his body, right, like chest, arms, legs, those high surface area body parts are smaller, right? So they have less sensory cortex. Okay. That is the homunculus. Don't forget his name. Okay. So fingers should have, you should be able to discern, okay, receptive field versus receptors versus cortex. So small receptive field for fingers, right? Because very, very sensitive. So we have 2, 4, and 5. If they're very small, that means there's a lot of receptors. So this one is out. So it's between 2 and 5. Now let's talk about cortex, large or small. For your fingers, they should have a large amount of sensory cortex. So it'd be 2. All right. So this is obviously not for you since you are not doing it in class. But you don't need to turn in your reflex results. Just need to be aware of the concepts for the exam. And then your pupillary reflex again, just try it out, right? It's probably good to check anyway. But I don't need to see that data. Just be aware of it for the exam and for the questions on the homework. And then you're just going to wait until Thursday when I post the data for every single section, and then you can complete your Lab 3 homework. Okay. So yeah, you'll complete Lab 3, and then of course pre-Lab 4 for next week. For you guys, okay, since we did not have in-person class, this is important and just for you. In order to receive your participation points, okay, since you did not attend in person, your attendance is based on watching this video. So once you finish this video, and only after you finish this video, right? And if you're already here, then hopefully you watched it all. You need to, at the end of your Lab 3 homework assignment, add a number 4 and answer this question here. So you should have your figure. There's only one figure this week, and then 4 questions. Okay. And you're turning that in, of course, like always, before the next lab. And that is all. So let me know if you have questions or concerns. If you're a little confused on these concepts, like you need to see them in person, we can chat about it before we start the next lab so you're caught up. All right. Thanks, guys.